The testis is surrounded by a dense fibrous capsule called the tunica albuginea, composed of collagen fibers that provide structural support to the testis. At the inner surface of the tunica albuginea, there is a plexus of blood vessels held together by delicate areolar tissue. This layer is called the tunica vasculosa. The tunica albuginea extends inward, forming septa that divide the testis into lobules. Each lobule contains 1 to 4 coiled seminiferous tubules. The seminiferous tubules are the functional units of the testis responsible for spermatogenesis. They are long, convoluted tubules lined with several layers of cells. The epithelium of each seminiferous tubule exhibits distinct characteristics due to the different stages of spermatogenesis in different tubules. Within the seminiferous tubules, you can identify the following cell types. At the basal compartment of the seminiferous tubules, we find spermatogonia, the stem cells responsible for sperm production. Spermatogonia appear as small, round cells with a large nucleus. They undergo mitotic divisions to produce primary spermatocytes with a prominent large nuclei. Moving towards the lumen of the tubules, primary spermatocytes enter meiosis I, leading to the formation of secondary spermatocytes. These cells then divide further through meiosis II to form round spermatids. The transition from spermatocytes to spermatids is including condensation of chromatin and the reduction of cell size. As spermatids progress towards maturity, they undergo complex morphological transformations during spermiogenesis. This process involves the elongation and differentiation of spermatids into spermatozoa, including shedding of their cytoplasm and development of a tail. At first, the spermatids have round nuclei, which undergo changes, becoming smaller, denser, and taking on the shape of sperm heads. These small black structures are the heads of fully mature spermatids or spermatozoa. Spermatogenesis is supported by Sertoli cells and Leydig cells. Sertoli cells are columnar-shaped cells that span from the basal to the luminal surface of the seminiferous tubules. They provide physical support and nourishment to developing sperm cells. Sertoli cells have elongated nuclei with prominent nucleoli. Leydig cells, also called interstitial cells, are found in the interstitial spaces between the seminiferous tubules. Peritubular myoid cells surround the seminiferous tubule and, together with Sertoli cells, form the cellular boundary of the spermatogonial stem cells. Interstitial connective tissue also contains fibroblasts, blood vessels, and nerves. The epididymis is a single-coiled tubule composed of pseudostratified epithelium with long immotile microvillia called strocelia. The epithelium also contains basal cuboidal stem cells. Beneath the epithelial layer, there is a thin layer of smooth muscles known as the muscularis. The thickness of muscularis layer increases from head to tail. In the head and body, there is a circular layer of muscle. The tail consists of 3 layers of muscles: an inner longitudinal layer, a middle circular layer, and an outer longitudinal layer. Surrounding the entire epididymis, there is a fibrous connective tissue layer called the tunica albuginea, which provides structural support to the organ.